Hello, and welcome to our video on gene expression. The expression of genes is accomplished through gene regulation. Gene regulation is when organisms can turn genes off and on when they need to. For example, in bacteria, gene regulation allows bacteria to have flexibility by turning genes on and off in response to environmental changes. If their environment lacks a specific amino acid that they need, for example, then they can turn genes on that produce enzymes to make that amino acid. Gene regulation is also important to eukaryotic organisms, like humans too. For example, every cell in your body contains the same DNA sequence, but some genes are turned on or off to create the components of a neuron, while other genes are turned on or off to create a muscle cell. When learning about gene regulation, there are some keywords to know. Structural genes encode proteins that are used in metabolism or biosynthesis, or that play a structural role in the cell. On the other hand, the products of regulatory genes, either RNA or proteins, interact with other DNA sequences and affect the transcription or translation of those sequences. In many cases, the products of regulatory genes are DNA-binding proteins, or RNA molecules that affect gene expression. Bacteria and eukaryotes use regulatory genes to control the expression of many of their structural genes. However, a few structural genes, particularly those that encode essential cellular functions, often called housekeeping genes, are expressed continually and are, therefore, said to be constitutive. Constitutive genes are not regulated. We will also encounter DNA sequences that are not transcribed at all, but still play a role in regulating genes and other DNA sequences. These regulatory elements affect the expression of DNA sequences to which they are physically linked. The regulation of gene expression can occur through processes that stimulate gene expression, termed positive control, or through processes that inhibit gene expression, termed negative control. Bacteria and eukaryotes use both positive and negative control mechanisms to regulate their genes. Looking at the figure, we can see that the DNA changes to RNA through the process of transcription, and then changes to proteins through the process of translation. In any of these steps, or intermediate stages, gene regulation can happen to affect the expression of that gene. Functionally related genes in bacterial cells are frequently clustered together in a single transcription unit termed an operon. A typical operon includes several structural genes, a promoter for those structural genes, and an operator to which the product of a regulator gene binds. The organization of a typical operon is illustrated in this figure above. At one end of the operon is a set of structural genes, shown as a gene A, gene B, and gene C. These structural genes are transcribed into a single mRNA, which is translated to produce enzymes A, B, and C. These enzymes carry out a series of biochemical reactions that convert precursor molecule X into product Y. The transcription of structural genes A, B, and C is under the control of a single promoter, which lies upstream of the first structural gene. RNA polymerase binds to the promoter and then moves downstream, transcribing the structural genes. Notice on the left-hand side of the figure, there is a regulator gene. A regulator gene helps control the expression of the structural genes of the operon by increasing or decreasing their transcription. Although it affects operon function, the regulator gene is not considered part of the operon. The regulator gene has its own promoter and is transcribed into a short mRNA, which is translated into a small protein. This regulator protein can bind to a region of the operon called the operator and affect whether the transcription can take place. The operator usually overlaps the end of the promoter and sometimes the end of the first structural gene. Gene expression in eukaryotes is regulated by different methods when compared to bacteria. One way of gene regulation is by changing the structure of the chromatins. Chromatin structure can be altered by chromatin remodeling complexes that reposition nucleosomes by modifications of histone proteins and by the methylation of DNA. Some transcription factors and other regulatory proteins alter chromatin structure without altering the chemical structure of the histones directly. These proteins are called chromatin remodeling complexes. They bind directly to the particular sites on DNA and reposition the nucleosomes, allowing transcription factors and RNA polymerase to bind to promoters and initiate transcription. Histone proteins are the globular proteins that DNA is wrapped around. Modifications in these histones affects gene expression by altering chromatin structure directly, or, in some cases, by providing recognition sites for proteins that bind to DNA and regulate transcription. Another change in chromatin structure associated with transcription is the methylation of cytosine bases. Heavily methylated DNA is associated with the repression of transcription in vertebrates and plants, whereas transcriptionally active DNA is usually unmethylated in these organisms. Abnormal patterns of methylation are also associated with some types of cancer. Transcription factors in eukaryotic cells can influence the initiation of transcription by affecting the stability or assembly of the basal transcription apparatus, or by recruiting coactivators that alter chromatin structure. Some transcription factors are activators that stimulate transcription. Others are repressors that inhibit the initiation of transcription. Some transcriptional regulator proteins bind to enhancers, which are regulatory elements that are distant from the gene whose transcription they stimulate. Insulators are DNA sequences that block the action of enhancers. Eukaryotic genes can be regulated through the control of mRNA processing. The selection of alternative splice sites leads to the production of different proteins. The stability of mRNA influences gene expression by affecting the amount of mRNA available to be translated. The stability of mRNA is affected by the cap, the poly-A tail, the UTR, the coding region, and the sequences within the UTR. RNA interference is initiated by double-stranded RNA molecules that are cleaved and processed. The resulting siRNAs, or miRNAs, combine with proteins to form complexes that bind to complementary sequences in mRNA or DNA. The siRNAs and miRNAs affect gene expression by cleaving mRNA, inhibiting translation, or altering chromatin structure. The term epigenetics comes from the Greek root epi, meaning over or above. The term epigenetics has come to represent the inheritance of variation above and beyond differences in DNA sequences. Epigenetics usually refers to the phenotypes and processes that are transmitted to other cells, and sometimes to future generations, but that are not the result of differences in the DNA base sequence. Many epigenetic effects are caused by changes in gene expression that result from alterations in chromatin structure or other aspects of DNA structure, such as DNA methylation. Some have broadened the definition of epigenetics to refer to any alteration of chromatin or DNA structure that affects gene expression. Here, we will use epigenetics to refer to changes in gene expression or phenotype that are potentially heritable but that do not alter the underlying DNA base sequence. Many epigenetic changes are stable, persisting across cell divisions or even generations. However, epigenetic alterations can also be influenced by environmental factors. Examples of environmental factors include nutrition, physical activity, air pollution, and stress. The fact that epigenetic traits may be induced by environmental effects and transmitted to future generations has been interpreted by some to mean that, through epigenetics, genes have memory. That environmental factors acting on individuals can have effects that are transmitted to future generations. Thank you for watching, and have a great day.